Good morning, good afternoon, and good evening, my friends in US, Europe, Israel, Asia Pacific, and Indian subcontinent. I welcome you all to Part 1 ASH 2021 Cellular Therapy Summary. So today we are going to review ASH CAR-T presentation. This is going to be a long session. If you get tired, recorded session will be available on IACH website. You can come back at any time to review abstract with original slides. To save time and to review more abstract, I'm going to speak about what is already, I'm not going to say much about which is already known to all of us. Special thanks to our friends, colleagues, and well-wishers who provided their slide to discuss their data with you all today. I have no conflict of interest to disclose, and I just wanted to mention I don't have a single stock of cellular therapy manufacturing companies, which we are going to discuss today. So we'll start with Allocarity Abstract first. We will begin with Alpha 2 study, Allo501, Allocarity in diffuse large B-cell lymphoma. Updated result continue to show encouraging safety and efficacy with consolidation dosing. Allogenic off-the-shelf CAR T-cell therapy addresses the logistic challenges, availability, and variable product quality of autologous CAR T we see. Allo501 is a genetically modified anti-CD90 Allocar T product that uses TALEN gene editing and permit the use of Allo647, which is an anti-CD52 monoclonal antibody like CANFET for selective and transitory host lymphodepletion. Author presented an update on preliminary safety, efficacy, and correlative data. Allo501A uses Selectis technology, as you know. This is a single-arm, open-label phase 1-2 trial with relapsed refractory large B-cell lymphoma, including relapsed refractory diffuse large lymphoma, follicular transformation, marginal zone lymphoma, primary mediastinal lymphoma, follicular lymphoma grade 3B, who received at least 2 or more lines of therapy. Subject with donor-specific antibodies were excluded. This is very important. Following lymphodepletion with Allo647, flu and Cy, either a single dose, 40 milligram, 40 million, or 120 million viable CAR T cell, or consolidation of Allo501A are given, dose level 2. Patients with stable or better disease control at day 28 receive consolidation with Allo647, no chemotherapy for lymphodepletion, and Allo501A dose level 2 cell infusion. 97% of enrolled patients initiated treatment with a median time from enrollment of only 2 days. You can see here all patients were heavily pretreated with advanced stage disease. Safety profile was manageable in both single dose and consolidation cohort. Event of interest in the single dose cohort have been previously reported a few months ago in ASCO presentation. In the consolidation cohort, CRS, no GVHDs, no ICONs, no dose limiting toxicity, no dose reduction, and no grade 3+ infectious complication occurred. That's fantastic. Of the both overall response rate and complete response rate were 50%. In the consolidation cohort, both overall response rate and CR rate were 60%, with all partial responses converting to CR after consolidation. Also, responses were ongoing. 6 of 7 patients who achieved CR remain in CR, and longest ongoing CR at present is month 15. As I mentioned, 3 PR converted to CR in consolidation 1, dose level 2. CAR T expansion was greater in patients who achieved a CR compared to patients who did not achieve CR. Patients who received the consolidation dose continued to have a CAR-T expansion after second CAR-T infusion. Amazing. So in summary, ALLO-501A with consolidation dosing demonstrated comparable safety and improved efficacy profile compared to single dosing, with all patients who received consolidation remaining CR. Let me clarify some regulatory issue in the past. In October 7, 2021, last year, FDA placed a clinical hold on Alpha-2 trial. I'm sure you are about it. After a case of chromosomal abnormality was reported in a single patient. The good news is, 3 days ago, on January 11th, or 5 days ago, January 11th, Allogene Therapeutics has been cleared to resume clinical trial work after US regulatory determined its technology was not at fault for an observed chromosomal abnormality in a patient. Nice abstract presented by our well-wisher, Dr. Jain. Precision Bioscience product. Investigator reported preliminary safety, efficacy, and correlative data for relapsed refractory B-cell ALL. Patient dosed with at least 3 million CAR-T cell per kilogram, allogenic, off-the-shelf, CD19-directed CAR-T. Derived from healthy donor cells, mitigate GVHD by knocking CAR-T directly into Treg locus. Subjects were 18 years or older with CD19 relapsed refractory B-ALL after at least 2 prior lines of therapy. Prior allo and prior auto CAR-T were allowed. Subject received either standard lymphodepletion with Flu/Cy or enhanced lymphodepletion, higher Flu/Cy dosing, 30 milligram per meter square 4 days flu and 1000 milligram per meter square Cy for 3 days, preceding PCAR 0191 infusion. As you can see here, dose level 3A and 4 combined due to lack of expansion upon second infusion without lymphodepletion in split dosing cohort. Median time from screening completion to product infusion of 7 days, and all eligible patients received CAR-T infusion. As of August 12, 2021, when this was presented, 15 patients with relapsed refractory CD19 B-ALL have been dosed with dose level of 3, 4A, or dose level 4B, flat dose of 5 into 10 to the 8 CAR-T cell. Patient characteristics presented in the table for your later review. Bottom line, all heavily pretreated patients. Most of the adverse events reported were mild, no cases of GVHD, no grade 3 or higher CRS, 1 case of grade 3 ICANS, which resolved within 48 hours. There was lymphodepletion. There was lymphodepletion standard versus enhanced lymphodepletion and CAR-T dose dependent changes in CAR-T expansion. Nicely presented table and figure here. CR or CRI rate at day 30 or beyond was 33% in dose level 3, 4A and standard lymphodepletion. And 80%, yes, 80%, 4 out of 5 patients in dose level 3, 4A with enhanced lymphodepletion, and 75% in dose level 4B, which was 8 into 8. Let's look at the dosing. 5 into 10 to the 8, higher dosing. %, 3 out of 4 in dose level 4B, 5 into 10 to the 8 with standard lymphodepletion. Amazing result. Importantly, 4 out of 15 patients, meaning about 25%, responding subject underwent allotransplantation, with one additional subject not able to receive transplant due to eligibility, yet he remained in the molecular MRD negative CR for now more than 250 days, and one refusing to proceed with transplantation. So in conclusion, this precision bioscience product, PB CAR 0191, has demonstrated a manageable safety profile and high complete response rate at day 28 or beyond, which provide an adequate window for potential bridging allotransplant in this advanced BALL. So now let's review the UCAR-T22 data. It is Selectis product. Nice presentation, impressive Allocarity product. Author presented safety and anti-leukemic activity of UCAR-T22 after FCA, flucimab lymphodepletion regimen. As we all know, company's prior preliminary data where host T cell recovery was observed in all patients after lymphodepletion with FC only, potentially interfering with UCAR-T22 expansion, and that's why they added alemtuzumab to the lymphodepletion regimen, FCA, to improve host lymphocyte suppression. UCAR-T22 is a genetically modified alloCAR-T product manufactured from non-HLA-matched healthy donor. Donor-derived T cells are transduced using lentiviral vector to express the anti-CD22 CAR and are further modified using selective T-line technology like using allogen product to disrupt the T cell receptor alpha constant, TRAC, and CD52 gene to minimize the risk of graft versus host disease and allows use of anti-CD52 directed drug for lymphodepletion. Eligibility criteria listed here. After lymphodepletion with FCA, a patient receive a single infusion of UCAR-T22 at 1 million or 2.5 million cells per kilogram. You can say here, all patients heavily pretreated with advanced disease. As of October 1st, 21, 12 patients received lymphodepletion, 11 patients were treated with UCAR-T22, and 6 of whom received lymphodepletion with FCA. Overall, well tolerated, no dose-limiting toxicity, and no ICANS. Twofold increase in inflammatory cytokines, including IL-6, IL-10, gamma interferon, CRP, in 2 of 3 patients in FCA cohort with anti-leukemic activity. Host lymphocyte remained suppressed throughout the day 28 dose-limiting toxicity observation period for all 3 patients in FCA dose level 2 cohort, 2.5 million cells per kilogram, correlating with UCAR-T22 proliferation. Here you can see twice a week flow data from day plus onward in patients receiving FCA lymphodepleting regimen. Depletion of UCAR-T22 expansion in patients receiving FCA lymphodepletion regimen. Nice figure showing increase in inflammatory marker in patients with detectable UCAR-T cell expansion. So in conclusion, FCA lymphodepletion regimen was well tolerated and associated with improved host lymphocyte suppression and UCAR-T22 expansion. Study is currently open, enrolling patients at FCA with higher dosing level. Dose level 3, 5 million UCAR-T22 cells per kilogram. Another allogen study, Allo501, produced deep and durable response in patients with relapsed refractory NHL comparable to autologous CAR-T. I'm not going to go into discuss, we already discussed before in brief, in short, allo off-the-shelf CAR-T cell therapy addresses our major logistic challenges, availability, variable product quality of auto CAR-T therapy. Allo501 is genetically modified anti-CD19 allocarity that uses talent gene editing technique, editing, selective technology, and permit the use of Allo647, an anti-CD50T monoclonal antibody for selective and transitory host lymphodepletion. Author presented an update on preliminary safety, efficacy, and correlative data. Fairly well tolerated, no dose-limiting toxicity, no GVHD observed. Overall response for autocar-naive patient by disease subtype and dose regimen summarized in this table. Excellent table for your later review. Objective response and complete response were 75% and 53% respectively. Impressive data. And it's ongoing. Durable response is observed. In conclusion, allogenic anti-CD19 CAR-T therapy can be safely and effectively delivered to patients with relapsed/refractory non-Hodgkin's lymphoma with encouraging durability of response. Now let's review some interesting Allocarity abstract. Let me start with this provocative ZUMA-12 data. First line axial cell therapy in high-risk large B-cell lymphoma. High-risk or ultra-high-risk large B-cell lymphoma is associated with poor prognosis. And we all know there is an urgent need for novel treatment for this high-risk, ultra-high-risk group of patients. So in this study, investigators reported the primary analysis of ZUMA-12, a phase 2 multi-center single-arm study of axi-cell as a part of first-line therapy in patients with high-risk large B-cell lymphoma. As eligible adult with high-risk large B-cell lymphoma, defined here, histology, double hit, triple hit, or IPI score of more than or equal to 3, plus positive interim PET scan per Lugano classification, dual score 4 or 5 after 2 cycles. For details, you can review Dr. Jason's JCO paper on score and classification. Patient underwent leukapheresis and received lymphodepleting chemotherapy, commonly used as nowadays conditioning chemotherapy, followed by single axi-cell infusion at 2 million cells per kilogram. As of May 17, 2021, 42 patients were enrolled and 40 treated with axi-cell first line in high-risk large B-cell lymphoma. Median age was 61, almost all super high-risk disease status to me. Look at here. Historically, I would say extremely poor prognostic group of these patients. Among all treated patients, overall response rate was 90%, CR rate was 80%, very impressive result in this ultra high-risk patient population. This is one of the GRACE case report, very advanced disease, achieved CR as early as 3 months after CAR-T infusion. You can see in here, durable response in most patients. I love this graph. 1 year overall survival, 90%, 91%, progression-free survival, 75%. All high-risk group. CR rate was consistent among key subgroup, double hit, triple hit, dual 5-point scale 4 or 5, and other subtypes listed here for your review. Grade 3 or higher CRS occurred in 3 patients, 8%, and no grade 4, no grade 5 CRS occurred. Grade 3 or higher neurological events occurred in 23%, but no grade 3-5 neurological event occurred. Higher frequency of naive T cell in ZUMA-12 compared to ZUMA-1. I mean CCR7 positive, CD45RA positive cell. Axi-cell was successfully manufactured in all 42 enrolled patients with median turnaround time of 18 days, very decent between leukapheresis and delivery to the trial site for treatment of patients. Median tumor burden appeared to be lower in ZUMA 12 than ZUMA 1 cohort, as expected, because ZUMA 1 is a first-line CAR-T trial. CAR-T expansion appeared greater in ZUMA 12 compared to ZUMA 1. Median time to peak level of CAR-T cell in blood was 8 days for ZUMA 12. Good news is that PK profiles were similar in patients with double hit or triple hit lymphoma and large B-cell lymphoma with IPS score of 3 or more. In conclusion, ZUMA 12 is the first study evaluating CAR T-cell therapy as a part of first-line therapy in high-risk or ultra-high-risk large B-cell lymphoma. Axi-cell demonstrated a high rate of rapid and durable response in patients with unmet medical need. I love this US Lymphoma Consortium group. A group includes all my favorite clinicians. Our center is part of the consortium. Brexucell, Tecartus for relapsed refractory mantle cell lymphoma, real-world experience from US Lymphoma CAR-T Consortium. We all know that Tecartus, Brexucell, was recently approved for relapsed refractory mantle cell lymphoma based on ZUMA-2 study data. Investigators in this study reported the pattern of use, safety, efficacy of Brexucell in standard of care practice among centers in US Lymphoma CAR-T Consortium. 14 centers participated in the study listed here. We are one of them. 95 patients received CAR-T infusion, most as a standard of care, very few on expanded access program due to slightly out of specification of product, I guess. The incidence of CRS and ICANS were comparable to those reported in ZUMA-2. Tocilizumab and steroid use appeared to be more frequent in this consortium study cohort. Almost 1/4 of patients required ICU transplant, 10 required ventilator, and 4 patients required mechanical ventilation. So incidence of CRS and ICANS were comparable to those reported in ZUMA-2. Tocilizumab and corticosteroid use appear to be more frequent in this consortium's cohort. Almost 1/4 of patients required ICU transport, as I said, and test patients required vasopressor, and four patient mechanical ventilation. A best overall response rate was comparable to that for reported in ZUMA-2 data, 90% range. Median time to initial response was 30 days. Day 30 overall response was 89%, including 66% CR and 22% PR. Nice graph. 6-month duration of DOR was comparable to that in ZUMA-2 studies. 12-month progression-free survival in this standard of consortium analysis was 51% versus 61% in ZUMA-2 study. And 12-month overall survival in this study was 72% versus 83% in ZUMA-2. Someone will question why this when we use as a standard of care lower response. And these are some of the explanation. More patients with intermediate, high-risk, simplified MIPI, blastoid, pleomorphic variants, T53 alteration, and more line of prior therapy in this consortium study compared to ZUMA-2. Now, if you look at more in detail, author presented nice data. 78% of patients analyzed here would not have met ZUMA-2 eligibility criteria. Despite all this, only clinical outcome is great. Let's move on to next abstract. My favorite abstract. First author is from Vanderbilt. Prophylactic corticosteroid use with Axi-cell. 1-year follow-up of ZUMA-1, cohort 6. ZUMA-1 is a phase 1-2 study of Axi-cell in patients with refractory large B-cell lymphoma. In ZUMA-1, cohort 1 and 2, 101 patient, data published in New England Journal of Medicine by Dr. Nelopu in 2017. Zuma-1 safety management cohort, which is cohort number 6, assessed whether prophylactic and early corticosteroid or tocilizumab could reduce the incidence and severity of CRS and neurological event. With median follow-up almost 9 months, 40 patients for cohort 6, there were no grade 3 or higher CRS, low-grade neurological and no low rate of severe neurological event, and high response rate. Paper was published in BJH last year, I think June 2021, by Oluwole et al. Here in ASH, investigator presented 1 year updated analysis of cohort 6, supported by propensity score matching PSM analysis to compare outcome for patients with cohort 6 and cohort 1 and 2. So 40 patient versus 101 patient comparison. This is a Zuma-1 study designed for later review. Cohort 1 and 2 versus cohort 6 who received prophylactic steroid. Why differences in terms of adverse event management between both groups? What was the difference? The briefly patient in cohort 3 received 1 daily oral dexamethasone dose, 10 milligram, on day 0 before XSL infusion, and also received on day +1 and day +2. Corticosteroid and tocilizumab were started earlier in cohort 6 than in cohort 1 and 2 for toxicity management, and it's reviewed here in detail. So no grade 3 or higher CRS occurred in cohort 6 versus 13% in cohort 1 and 2. Grade 3 or higher neurological events were in only 15% versus 28% in cohort 1 and 2. Median DOR, PFS, overall survivors were not reached yet. Investigators said overall response rate remained 95%, 80% CR, with median follow-up of 15 months. At data cutoff, more than half of patients were in an ongoing response. Then investigator presented the propensity score match analysis. In total, 32 match patients, each in cohort 6 and cohort 1 and 2 were identified in this PSM analysis. Clinical efficacy remained comparable between patients in cohort 6 and cohort 1 and 2 before and after propensity score matching. You may come back to review this slide later. It will be on IACH website. So incidence of grade 3 or higher CRS was lower in cohort 6 compared to cohort 1 and 2 before and after PSM. Median time to onset of any grade CRS was delayed in cohort 6 versus other cohort before and after, and the difference was 2 days versus 5 days after CAR-Ts. So you can start safely outpatient CAR-T infusion. Median time to onset of grade 3 or higher neurological event appeared to be delayed in cohort 6 versus cohort 1 and 2 before and after propensity score match analysis, 7 days versus 12 days after CAR-T. And this is very interesting. Despite using prophylactic steroid for 3 days, median cumulative corticosteroid dose, including prophylaxis, was 6-fold lower in cohort 6 versus cohort 1 and 2, 1200 milligram plus versus 7 gram plus after matching. Peak CAR-T level were comparable, and peak inflammatory biomarkers associated with CAR-T cell treatment-related adverse events were lower in cohort 2 as expected versus cohort 1 and 2 before and after matching. And it supports clinical outcome also. So in conclusion, I don't think I need to read this slide, but cohort 6, and that's what we practice in our institution. It works out very well without compromising outcome. So this is one more CAR-T consortium study. Outcome of aggressive B-cell lymphoma, a patient with no evidence of measurable disease at the time of CAR-T therapy, experience from CAR-T consortium. And this happens, patients get into CR, especially after bridging therapy while awaiting CAR-T spot, manufacturing, or infusion. And these are the group of patients they analyzed. That's what this analysis is about. Tumor burden determines CAR-T outcome. If you remember in the Juliet trial, 7 patients without disease at infusion, 5 remain in CR at 12 months post-transplantation. CRS, grade 3 was only 28%, ICONs 14%, all grade 1. So that's what I know about tumor burden. As I said, in Juliet study, 7 patients without disease at infusion, 5 remain in CR at 12 months post-transplant. ICONs was very low, all grade 1, 14%. Low toxicity with no disease burden. Study population in this analysis included patients with no evidence of measurable disease at the time of CAR-T infusion. Total 15 patients with diffuse large B-cell lymphoma without evidence of disease at the time of CAR-T infusion. Please don't question why they gave CAR-T infusion in these patients. We already discussed before. They were all eligible for CAR-T. 13 of 15 patients received bridging treatment and attained CR. None developed severe CRS and none developed any ICONs. Yes, 0% ICON. 1 year overall survival, 73%, event-free survival, 66%. Pretty long, good follow-up. So in conclusion, patients with diffuse large B-cell lymphoma who had no residual disease at the time of CAR-T infusion had excellent outcome. Remember, they were all eligible for CAR-T when they were referred to CAR-T. Patients with low or no disease burden may be managed in an ambulatory setting. My favorite group at UCL, Claire, Carl, and others, abstract was presented by Claire. This was presented as a poster. You may come back to review poster later on IACH website, but let me briefly review the data. Auto1, Obe-cel is an autologous CD19 CAR-T product engineered with fast off-cell CAR to reduce immune toxicity, improve CAR-T engraftment. Author previously published data cited in the poster. In short, previous data confirmed that Auto1 is associated with low level of CRS, durable CAR-T engraftment, high MRD negative CR rate in adults with relapsed refractory B-cell ALL. Claire is the first author. Paper was published in JCO 2021. So in this study, author presented Auto1, Obe-cel, initial data in indolent and aggressive relapsed refractory B-cell NHL and relapsed refractory B-cell CLL. A classical study design, I'm not going to go into detail, but subject more than 16 years or higher older, lymphodepletion with flu-cy prior to Obe-cel infusion, with exception of diffuse large B-cell lymphoma, who additionally received a single dose of pembrolizumab on day -1 to potentiate CAR-T expansion. Auto1 dose varies based on the indication. Split dosing means 200 and 30 million CAR-T at day 0 and day 9 in CLL cohort, and single dose of 200 million CAR-T in patients with relapsed refractory B-cell ALL. Consort diagram and baseline characteristics summarized here. Apheresis and product manufacturing was successful in all patients. No grade 3 or higher CRS, no ICONs observed in this study. Submitted abstract mentioned 1 patient, a partial patient developing macrophage activation syndrome, which resolved with anakinra and steroid. At median follow-up of 6 months, 8 of 9 patients were disease-free at last follow-up. Very impressive data like their CLL, like their ALL data. Study is still ongoing. More exciting data to come. T-cell. A T-cell versus versus standard of care as a second-line therapy of primary refractory relapse aggressive B-cell non-Hodgkin's lymphoma. This is a phase 3 Belinda study data. We will review several other abstracts comparing CAR-T with standard of care in second-line therapy for relapse refractory large B-cell lymphoma. This particular abstract was presented in late-breaking abstract session by my favorite attending when I was working as a fellow at NIH, and he was at the NIH at that time, Dr. Bishop. So Belinda is a global phase 3 randomized trial comparing the efficacy and safety of T-cell to the current standard of care second-line therapy strategy in patients with relapsed refractory aggressive non-Hodgkin's lymphoma within 12 months of first-line therapy. This is a classical study design. Adults with confirmed relapsed refractory non-Hodgkin's lymphoma within 12 months after first-line chemoimmunotherapy were eligible. Arm A received optional bridging treatment, investigator choice of protocol defined as a platinum-based chemoimmunotherapy and T-cell infusion. Arm B received investigator choice platinum-based chemoimmunotherapy regimen followed by auto-transplant in responder or second-line platinum-based immunochemotherapy in non-responder. Crossover was allowed if no response at week 12 by blinded independent reviewer committee until 1 year post-auto-transplantation. Slightly stringent criteria for crossover. Baseline characteristics summarized here for later review. Clinical trial flow is reviewed here for your reference. Median time to infusion for all patients on T-cell arm was 52 days, which is pretty long time. In the US center, it was 41 days versus 57 days in non-US center. I would say way longer than I would expect in this group of patients to give them help. Slide reviewed overall response rate week 6 and best overall response. Very nicely summarized here. And there was no difference in event-free survival between arm A and arm B. 32% in T-cell and 28% in standard of care patient, standard of arm died on study, mostly as expected due to progressive disease. So in conclusion, message is event-free survival was significantly different between T-cell and standard of care treatment strategy in patients with aggressive non-Hodgkin's lymphoma that was refractory or relapse early after first-line therapy. Their finding was also suggested the importance of preventing progressive disease prior to infusion, almost 50 days before they received the CAR-T infusion. Higher proportion of patients had progressive disease at week 6 prior to CAR-T infusion. That makes sense. In this T-cell arm, and we already reviewed median time for infusion. It was almost 52 days. So effective bridging prior to CAR-T infusion and shorter time to infusion for this chemotherapy refractory patient could be critical to improve outcome. Dr. Bishop also mentioned that additional analyses are ongoing to understand the result. This is the recent publication in New England Journal of Medicine. I'm sure you are all aware about a recent competitive study by Kite Gilead, ZUMA-7 data, also published in New England Journal of Medicine recently. Let's switch to BMS product, Lysocell. Manali, Dr. Kamdar presented similar abstract, but using Lysocell, a BMS product. Patient with large B-cell lymphoma, primary refractory or relapse less than 12 months after first-line therapy has a poor outcome with standard of care. We all know that. I don't need to review prior Lysocell publication, their result. In this study, author presented interim analysis of transform study evaluating the efficacy and safety of lysocell versus standard of care as a second-line therapy in patients with relapsed refractory large B-cell lymphoma, similar to study presented by Dr. Bishop, but using Novartis product. The Transform is a pivotal global randomized multi-center phase 3 study comparing efficacy and safety of standard of care. Standard of care, arm A, they use DHAP, RDHAP, RICE, RICE, RGDP salvage regimen, personal investigator choice, followed by beam and auto transplant, or arm B, which is a lysocell. Again, crossover to lysocell was allowed, but slightly liberal criteria, different crossover criteria compared to previous presentation we discussed before. A total of 184 patients randomized with 92 patients in each arm. Baseline characteristics were well balanced between both groups. Event-free survival was significantly longer, and CR rate was significantly improved for arm B, lysocell arm, versus standard of care arm A. Overall survival data were immature at the time of this analysis with median follow-up of 6 months, but numerical trend favored the blue line, which is a lysocell arm. Only one patient had grade 3 CRS and onset of at day 9, which resolved in 2 days. Grade 3 neurological events was only in 4%, and no grade 4, grade 5 events occurred. Persistence of lysocell was observed up to 12 months after infusion with follow-up ongoing. In the lysocell arm, more than 95% of patients had B-cell aplasia pre-infusion and at 1 and 2 months after infusion. So similar to ZUMA-7, but in contrast to a phase 3 Belinda study, this study showed benefit of lysocell over standard of care with highly significant and clinically meaningful improvement in event-free survival, CR rate, PFS as second-line therapy in patients with large B-cell lymphoma, refractory or relapse within 12 months after first-line therapy. Again, all studies have some differences in selection criteria, so I would not say that one product is better than other product. ZUMA-5. Long-term data was presented by our friend Dr. Nilapu. As you know, ZUMA-5 is a multi-center, single-arm phase 2 study of axi-cell in patients with relapsed refractory indolent non-Hodgkin's lymphoma, including follicular and marginal zone lymphoma. This is a study design. Adult patients with relapsed refractory follicular or marginal zone lymphoma after 2 or more lines of therapy, leukapheresis, conditioning chemotherapy, followed by axi-cell infusion, 2 million cells per kilogram. Overall response rate was 94%, 79% CR among follicular group, 83% overall response in marginal zone group, 63% CR. At data cutoff, 57% of efficacy eligible follicular lymphoma and 50% marginal zone lymphoma had ongoing response. Of patients who achieved CR, we are almost half. Of patients who received CR, 68% of patients with follicular lymphoma and 73% of with marginal lymphoma, small numbers, had ongoing response. Almost 2/3 of all CR patients had ongoing response. That's a good news. 2-year event-free survival for follicular lymphoma, 63% and 47% for marginal zone lymphoma. Median overall survival was not yet reached in efficacy eligible patients in both follicular and marginal zone lymphoma group. Long-term PFS rate in patients with follicular lymphoma were generally consistent among key subgroup. Slide here for your review. Among marginal zone lymphoma, again same, despite limited sample size, progression-free survival at 12 months appeared to be consistent among key subgroups, including patients who had prior treatment with and failure with BTK inhibitor. Quite safe. I don't think I need to go into the details. CAR-T expansion, very important by peak, and AUC was significantly higher in follicular lymphoma patients who had ongoing response at 12 months post-infusion than those who were relapse. Pharmacokinetic findings were similar in patients with marginal zone lymphoma. Slide nicely summarized the detectable B cell and CAR T cell over time in patients with follicular lymphoma and ongoing response at 24 months. In conclusion, as you know, Kexicel is FDA approved for our adult patients with relapsed refractory follicular lymphoma after 2 or more lines of therapy. This indication is approved under accelerated approval based on response rate. Continued approval for this indication may be contingent upon verification and description of clinical efficacy in confirmatory trial. In terms of marginal zone lymphoma, ZUMA-5 data showed efficacy. Outcome appear to be improved with long-term follow-up, but not yet approved by FDA. Next abstract, analysis of outcome and predictor of response in patients with relapsed mantle cell lymphoma treated with Brexucel, presented by Jason from Emory in Atlanta. We want to know what's going on, right? As you know, the pivotal ZUMA-2 trial demonstrated an overall response rate of 85%, complete response rate of 59%, and 1-year progression-free survival of 61% in mantle cell lymphoma patients treated with Brexucel, also called TCARTers. Author conducted multi-center prospective study to assess the safety and efficacy of Brexucel on a non-trial setting. It looks like I have some problem with intervals. It gets jammed sometime. My apology. This was a retrospective review of records across 14 academic medical centers in United States. 69 patients underwent leukapheresis. 66 patients ultimately went to receive the infusion of Brexucel. This is characteristic summarized here. Table summarizes the prior therapy in patients who received Brexucel. All heavily pretreated advanced disease feature. In terms of outcome, objective response was 86%, complete response rate was 77%. With a median follow-up of 4 months, estimated 6-month progression-free survival was 77%, and 6-month overall survival was 88%. In terms of adverse event, 12% with grade 3 or higher CRS, 1 patient died as a result of CRS. 35% experienced grade 3 or higher ICANS. There was 1 grade 5 events. This is very important. Among 8 patients with history of CNS involvement prior to Brexucel, 5 experienced ICANS with maximum grade of 2 in 1, grade 3 in 3, and grade 4 in 1 patient. But the good thing is, all 8 patients are alive and free of relapse at last follow-up. Let's switch gear to, I don't think I need to read the conclusion slide, that's why I skipped to save time. So long-term survival in ZUMA-1, pivotal study of Kexicel in refractory large B-cell lymphoma, presented by Karen from Dana-Farber Cancer Institute. I don't think I need to read the background on Kexicel in relapsed refractory lymphoma. It's here for your later review, case needed. The objective of analysis is to present 5-year updated analysis, including patients from cohort 1 and 2 from phase 2 ZUMA-1, and to explore potential role of event-free survival at 12 and 24 months as a surrogate endpoint for long-term survival. Very smart plan. We want to predict at 1 or 2 month post-transplant, 1 and 2 year post-transplant, if we can say patients are cured or not. Zuma-1 treatment schema is shown here for your reference. As previously reported, total of 101 patients received exa-cel, comprising the modified intent to treat population. In this 5-year analysis, median follow-up time was 63 months. I received a slide from Karen, but her recording is still there. So let me conclude this presentation. In this updated 5-year analysis, exa-cel induced long-term survival with 5-year overall survival rate of 43% among treated patients. Between 4 and 5-year analysis, time to next therapy curve remained stable. 92% of patients remained alive without need of subsequent therapy, which may be suggestive of cure of this patient. Exploratory analysis of long-term overall survival by event-free survival status appear highly correlative. So what we can say is event-free survival at 1 year and 2 years as a surrogate endpoint for long-term overall survival in relapsed refractory large B-cell lymphoma. Loved her data. Again, I received all slides from the speakers, and some of the slides I was not able to remove their audio recording. That's why sometimes we get jammed and some conflict when I present the data. So let's move on to next presentation. Outcome and approach who relapse after CD19 CAR-T present. So there will be a few abstracts on that. Nice abstract, CD22 CAR-T therapy, mediate high durable remission in adults with large B-cell lymphoma who have relapsed after CD19 CAR-T therapy. We all know outcome of CAR-T, CD19 CAR-T therapy. When they relapse after CD19 CAR-T, prognosis is very poor. Nicely summarized slide for your reference. Author previous data showed that treatment with CD22-directed CAR-T therapy produced 70% CR rate in 58 pediatric patients with heavily pretreated ALL. And this is an ongoing single institution phase 1 dose escalation clinical trial. Study design reviewed here, 2 dose levels for your reference. Study population included relapsed refractory large B-cell lymphoma, including secondary CNS involvement. 2 dose levels. Dose level 1, 10 to the 6, 1 million cells per kilogram. Dose level 2, 3 10 to the 6, 3 million CAR-T cells per kilogram. Heavily pretreated patient. All patients had at least one high-risk feature, including failure of prior CAR-T therapy, refractory disease to second-line or later therapy, and other features are summarized here. Only 1 patient had grade 3 CRS. No grade 3, 4 icons. All cases were grade 1, 2, resolved within 2 days. Interestingly, there is more. 5 patients, 21 patients experienced MAS, hyperinflammatory macrophage activating syndrome, manifested in all cases by severe pancytopenia, consumptive coagulopathy, DIC, requiring transfusion and growth factor support. So here are the clinical outcome of all patients and dose level. Outcome appeared to be similar between the dose cohort. Dose level 2 had lower overall survival compared to dose level 1 due to early death from infection. The combined overall response is 79%, CR 58% for first 24 patients enrolled. Complete response to CAR-22 appeared durable with only 1 patient who relapsed to date based on their presentation. Based on this data, investigators have chosen dose level 1 as a recommended dose phase 2 dose. And this is a nice review, superb review. And this is like a board question on macrophage activation syndrome after CD22 T cell expansion. 5 out of 24 patients and 3 out of 5 patients developing dose level 2. And that's why they selected probably dose level 1 for phase 2 study. In summary, infusion of CAR22 in relapsed refractory large B-cell lymphoma is safe and well tolerated at dose level 1. Manufacturing of CAR22 was uniformly successful. This data demonstrate CAR22 being an effective cell therapy for CAR19 refractory or CD19 negative large B-cell lymphoma. Safety and efficacy of FT596, first in class multi-agent, multi-antigen targeted, off-the-shelf, iPSC-derived CD19 CAR-T NK cell therapy in relapsed refractory large. This is a FATE therapeutic product. FT596 is an induced pluripotent stem cell, iPSC-derived, off-the-shelf CD19 CAR-T, CAR natural killer cell therapy, capable of multi-agent targeting in combination with monoclonal antibody therapy. Conditioning chemotherapy included Flu/Cy administered, followed by single dose of FT596 as a monotherapy in regimen A, or combined with rituximab, standard dose, 375 per milligram per meter square, regimen B1, or obinutuzumab, 1000 milligram per meter square, regimen B2 on day 4. Nicely summarized study design here for your later review. Patients experiencing clinical benefit may receive a second cycle of conditioning, followed by single dose reinfusion of FT596 with FDA approval in cycle 2. 2-dose treatment schedule on day 1 and day 15, initially initiated recently, so we don't have those data. I'm going to talk about A and B1. Slide review the patient and disease characteristic of both regimen A and regimen B1, Flu/Cy with R. All advanced disease, heavily pretreated, no dose-limiting toxicity reported with either regimen, no graft versus host disease or ICANS observed, and no severe CRS. Of the 17 efficacy eligible evaluable patients, 5 of 8 in regimen A and 4 of 9 in regimen B, which include rituximab, achieved objective response after first FT596 treatment cycle. Single dose level, more than 90 million cells, 8 out of 11 efficacy patients observed objective response, including 7 CR, 7 out of 11. Of 4 patients with prior CAR-T therapy treated with more than 90 million cells, 2 achieved CR, impressive data. FDA approved all requests for retreatment, with day 1 and day 15. Second cycle was well tolerated. 11 treated with second cycle, and clinical activity was maintained. 6 of 11 patients with CR, maintained CR, 4 patients pending cycle 2 assessment as data cutoff, and fantastic data from a FAT therapeutic product. Median study follow-up time for patients treated with more than 90 million drug is 4.2 months. 10 of 13 responders remain in response at data cutoff. In conclusion, FT596 exhibits clinical benefit in patients with advanced relapsed refractory B-cell lymphoma. One more good option, right? FT596, even in those patients who relapse after CAR-T or relapse after allotransplantation. This poster, I just plugged in poster for later review. It was presented as a poster presentation. Still very early, but it highlights Iovance's cellular therapy design. Next presentation, let's switch gear to CAR-T and IEC in other malignancies. Why not CAR-T for Hodgkin's patient? Nice abstract presented by Dr. Ahmed from the Anderson. CD19 CAR-T cell therapy in patient with relapsed refractory classical Hodgkin's lymphoma. Nice recent JCA paper cited here for your review. In two phase 1/2 clinical study, autologous CD19 CAR-T therapy was well tolerated with significant clinical activity in relapsed refractory classical Hodgkin's lymphoma. Here in this presentation, Dr. Ahmed presented result of pilot segment of phase 2 pivotal trial of autologous CD19 CAR-T in relapsed refractory classic Hodgkin's lymphoma. Classical study design, all patients progress after at least 3 lines of therapy, including chemotherapy, brentuximab, and PD-1 antibody. Previous auto or allo was allowed in this trial. Pivotal segment planned to enroll approximately 82 patients to obtain at least 67 CD3 CAR-T treated patients. Lymphodepleting chemotherapy used bendamustine and fludarabine, 3-day schedule highlighted here. Data reported 15 patients enrolled in pilot segment. 14 patients were eligible for treatment with CD30 CAR-T, heavily pretreated. CD30 CAR-T product was successfully manufactured for all patients with a median manufacturing time of 6.3 weeks. Bridging therapy was allowed. All 14 CD30 CAR-T treated patients were infused with planned dose. Median dose was 2.410 to the 8 cells per meter square. Fairly well tolerated, no fatal adverse event. Only 1 grade 1 CRS, no ICANS. Overall response at day 42 after CD30 CAR-T as assigned by investigator was 100%, 5 out of 5. Small study, but impressive data. CR in 80%, PR in 20%. Small number again, but very impressive. I like the data. Pharmacokinetic analysis of CD30 CAR is still ongoing, but you can see here expansion. Excellent so far in this study. Efficacy and safety of CD30 CAR-T will be further evaluated in pivotal segment of this phase 2 study. Next abstract, phase 1/2 trial of IL-7, IL-15 expanded bispecific LV2019 CAR-T cell for relapsed refractory B-cell non-Hodgkin's lymphoma. Abstract was presented by very smart guy, Dr. Nirav Shah. Bispecific lentiviral anti-CD20 and anti-CD19 called LV2019 CAR-T cell may improve outcome in relapsed refractory B-cell malignancies by limiting relapse from single antigen downregulation. Their initial publication, Shah et al., published recently in Nature Medicine 2020 for your reference. In this ongoing phase 1/2 clinical trial, his team examined the impact of IL-7/15 in lieu of IL-2 in their prior study and varying length of manufacturing, 12 days versus 8 days for patients with relapsed refractory non-Hodgkin's lymphoma, safety and efficacy study. Again, I'm not going to review detail. This is author's prior work on phase 1 trial published recently in Nature Medicine for your reference. CAR-T cells were locally manufactured in the clinic, Max Podigi device with IL-7/15 for cell expansion, with goal of fresh infusion at very length of manufacturing, like I mentioned, 8 or 12 days. In total, 22 patients received LV2019 CAR-T at a specified dose of 2.5 million cells per kilogram. In total, there was one dose-limiting toxicity in phase 1b, 12-day arm with prolonged 12-day arm, not 8-day arm, 12-day arm with prolonged grade 3 ICANS and grade 4 acute kidney injury. There were 2 non-relapse mortality outside the DLT period from infectious complication. CRS occurred in 19 patients, mostly grade 1 or 2, 18 out of 19. ICANS occurred in 27%. Again, 14% had a grade 3 ICANS. Median day to CRS was 1 day, which correlated with a rapid in vivo expansion of the cell. Among all patients, overall response rate was 91%, 55% complete response. 17 patients had MRD testing and 13 were MRD negative. Median follow-up of survival is 6 months. Median event-free survival and overall survival have not been reached yet today. 20 of 22 patients received fresh CAR-T without cryopreservation, and which saves lots of cost. Also, I know how much we are paying for cryopreservation. Outcome of diffuse large B-cell lymphoma patient. Summarized here for your reference. And this is outcome of mantle cell lymphoma patient. Excellent outcome, you can see here. So in conclusion, bispecific LV2019 CAR T-cell expanded with IL-7/15 are safe, efficacious for relapsed refractory B-cell NHL with high overall response rate, low level of severe CRS and ICANS, improved response among patients treated with short 8-day manufacturing process. Mantle cell lymphoma outcomes were impressive with 100% overall response rate and no relapse to date. How about CAR-T in relapsed refractory T-cell non-Hodgkin's, T-cell ALL, sorry. This abstract was presented by famous CAR-T program in China, led by Dr. Liu. CAR-T therapy in TLL is still being explored in early clinical stage trial. I'm sure you are all aware about recent JCO paper. In this study, author presented outcome from their phase 1 clinical trial of CD7 CAR-T therapy for relapsed refractory T-cell ALL. All patients received standard 3 days lymphodepleting chemotherapy prior to CAR-T infusion. Second generation CD7 CAR-T is composed of anti-CD7 single chain antibody. Nice illustration of their preclinical comparison of 3 CD7 CAR-T manufacturing method. 17 relapsed refractory T-cell ALL patients were enrolled between December 2020 and June 2021. Data from 14 patients is available for evaluation, all heavily pretreated, including 3 relapse after allotransplant. CD7 CAR-T were 100% successfully manufactured. 12 of 14 patients received bridging therapy. All by day 28 post-infusion, 13 out of 14 patients achieved CR, with all 13 of 13 patients with MRD negative CR or CRI. Consolidation allo was permitted at the treatment physician discretion and patient's preference. For 11 of 14 patients were bridged to consolidation allo at a median of 57 months, 2 months after CD7 CAR-T infusion. None of the 14 patients had grade 3 CRS, no severe neurotoxicity. Importantly, very important, CD7 CAR persisted well in peripheral blood at a median of 52 days at last evaluation, regardless of their transplantation status. Very impressive. In conclusion, data showed that CD7 CAR-T therapy is safe, highly effective in treated patients with heavily treated relapsed refractory T-cell ALL, including those with extramedullary involvement, history of prior allo or high-risk subtype. Many of these patients received second allo after CD7 CAR-T therapy. I like this product. MENA therapeutic product. Again, I don't have single stock in any industry product. Outcome of donor-derived tumor-associated antigen-specific T-cell therapy in patients with high risk or relapsed acute leukemia post-allo transplantation. And as a transplant physician, this is what we want to know. How can we treat post-transplant relapses or prevent relapses? So post-allo transplant relapse is a nightmare, especially early relapse. I don't think we need to review the literature. TAA, tumor-associated antigens, are potentially immunogenic, and tumor-associated antigen expression can induce a T-cell immune response. Next plan. To boost the GVL effect, infusion of donor-derived T-cell targeting tumor-associated antigen peptide as circulating TAA-specific T-cells are associated with maintenance of remission post-transplant, persistence of graft-versus leukemia effect. And so in this phase 1 study, author evaluated the safety and clinical outcome following administration of novel T-cell therapeutic targeting 3 TAA, tumor-associated antigen, WT1, PREM, and surviving in patients with acute leukemia who relapse or at high risk of relapse after allotransplantation. 23 patients with relapse refractory, 11 patients, or high risk for relapse, 12 patients, mostly AML, were infused post-transplantation. No major toxicity. One patient had only grade 3 GvHD, otherwise no issues. You can see here outcome of patient who received TAA T-cell infusion among relapse and adjuvant for high risk after transplantation. But the preemptive was very effective in high risk disease population and safe. Instead of DLI, preemptive TAA T-cell. Immunofluorescent expression of TAA target demonstrated on patients' blast population. Very nice figure. Durable response post-TAA T-cell infusion, tumor-associated antigen T-cell infusion in patients with high-risk disease treated preemptively. Nice figure. Ongoing response in most patients. Evaluation of unique T-cell receptor site clonotypes demonstrated expansion and persistent in patients up to 1 year post-infusion. Shown here again. In conclusion, although as a phase 1 study, concomitant anti-leukemic therapy was allowed, like we do with DLI, post-transplant DLI, chemotherapy, Vidaza combination for relapse or high-risk patient, TAL therapy was shown to be very effective and safe. Sustained remission observed in high-risk and relapse patients. Moreover, adoptive transfer TAA T-cell expanded in vivo as detected by sequential sequencing persisted up to 1 year post-infusion. And this is interesting abstract. Again, all of them are interesting. Primary progression on frontline therapy for diffuse large B-cell lymphoma is associated with poor outcome despite subsequent CAR-T therapy. We want to predict who will not get benefit from CAR-T, so we can be ready with plan B. Between 50 and 80% of patients with diffuse large B-cell lymphoma are cured by frontline R-CHOP chemoimmunotherapy. Ultra-risk feature for poor survival include progression through frontline therapy and many other features listed here. Authors aim to explore role of this ultra-high risk feature in outcome of diffuse large B-cell lymphoma patient receiving standard of care CAR-T therapy. We do give CAR-T therapy to everybody, all refractory. So we want to see how they did in ultra-high risk feature. So this is a retrospective single-center study, relapsed refractory diffuse large B-cell lymphoma treated with axi-cell or TISA cell, standard of care at Moffitt Cancer Center, large-sized CAR-T program, standard of care, or who were received expanded express CAR-T cell for provision of CAR-T when products fell outside the manufacturing specification. So primary treatment failure occurred in 75 patients, all high-risk future. Among patients with primary treatment failure group, 69 received axi-cell and 6 received T-cell, so majority axi-cell, and just generally preference at most programs. Median line of therapy prior to CAR T-cell therapy was 3 days, ranges from 2 to 7 days. Median overall survival for patients with 2-3 ultra high-risk features was 5 months versus not reached yet in patients with ultra high-risk feature of 0 to 1. Primary progression, meaning PP, meaning progression through the frontline therapy was associated with worse progression-free survival and overall survival after CAR-T compared to other pattern of failure. So ultra high-risk feature mentioned here is progression through frontline therapy, PP, presence of MIC translocation, MIC R+, and high or high intermediate NCC and IPI. So in conclusion, relapsed refractory diffuse large B-cell lymphoma patient with progression through frontline therapy present a poorest subgroup even with CAR-T therapy, and we need to be ready with plan B. I love this abstract. Dr. Radhika Bansal from Mayo Clinic presented this abstract, and I previously worked when I was fellow at IACH on lymphocyte subset recovery and transplant outcome. In this study, Radhika presented peak absolute lympho count post-CAR-T correlate with clinical response in aggressive lymphoma. Author hypothesized that initial lymphocyte expansion in first 2 weeks post-CAR-T infusion is due primarily to CAR-T expansion. So peak absolute count, absolute lymphocyte count, ALC peak, readily accessible by clinical CBC with differential and can be surrogate for CAR-T expansion. So they examined the ALC peak after CAR-T infusion and correlation with clinical outcome for NHL patient who received FDA-approved axi-cell at Mayo Clinic between 2016 and 2021. ALC was determined for differential white cell count. ALC peak was defined at the highest ALC value in any differential count in first 15 days post-CAR-T infusion. Median time to ALC peak was 10 days. Increasing ALC peak level was correlated with increased CAR-T in the peripheral blood by flow. Patients who achieved CR as a best clinical response or durable CR had a higher ALC peak compared to those who did not. Simple test to predict response. Using ROC analysis, with receiver operating characteristic analysis, ALC peak level of 670 was defined as a cutoff point for association with CR as a best response, 0.67. So high ALC peak was correlated with prolonged EM-free survival. Prognostic association remains statistically significant on multiple analyses also. Same as for overall survival. So in conclusion, authors have seen in a previous group, we know how to predict now based on simple formula, but we have also seen in previous presentation, progression through the frontline therapy as a prognostic marker for CAR-T. And now a low ALC peak in first 15 days correlated with CAR-T level and outcome. So groups we need to ready for next line of action to help patient. And there's one more abstract on impact on CAR-T outcome we'll be reviewing soon. And this is a nice CIBMTR study. We have recently published Nice review article on allo transplantation after CAR T in BGH for your reference. And this study shows clinical data. Despite the remarkable initial CR rate, many patients relapses after CAR T for relapse refractory B-cell ALL. A key remaining clinical question is the role of post-CAR T allo transplantation for salvage allo for relapse after CAR T or as a consolidation in remission after CAR T. So two things here we are analyzing. Allo transplantation as a salvage for patients who are relapsing after CAR T, and allo transplantation as a consolidation in remission after CAR T in patients who had relapsed refractory ALL. Patient up to 26 years treated with commercial or investigational CAR T cell product from 2014 to 2019 United States were included. CABMTR is our data source to study the impact of subsequent transplant following CAR T therapy. Nice summarized method. So in this study, one of the authors identified 347 patients who received CD19 CAR T from 86 centers reporting to CABMTR. Patient, and this is characteristic of both group reviewed here, both group meaning consolidation allo and salvage allo after CAR T. Of the 347 patients who received CD19 CAR T therapy, 18% had a subsequent transplant in CR as a consolidation post-CAR T, with median time from transplant from CAR T to transplant was 5 months. 36% patient had prior to, that means they had a second transplant, but the majority, because they are young patients, majority received ablative regimen and majority were MRD negative at the time of transplantation. Post-CAR T transplant used as a consolidation was associated with lower risk of relapse, but no significant impact on mortality and disease-free survival. Now, when accounting for any transplant performed after CAR T, transplant was associated with high mortality. Still these are young patients up to the age of 26. You can see here overall survival in patients receiving subsequent allo transplant with or without relapse after CAR T therapy. Nice figure. So we need to do it before they can relapse. So in conclusion, consolidation transplant after CAR T cell resulted in a significant reduction in leukemia relapse, which are trend toward better disease-free survival. Consolidation allotransplant appears safe, incurred a benefit by reducing the disease relapse. Overall survival in patients receiving subsequent allotransplant without relapse after CAR T therapy had a superior outcome. So we always talk about axi-cell or T-cell, in our discussion. I like this study. And they're comparing both axi-cell and T-cell propensity score match comparison of axi-cell and T-cell for relapse refractory large B-cell lymphoma in real life. A LISA study from Descartes registry. I don't think I need to review this data. Both approved CAR T product, and we have already reviewed several abstracts. Both pivotal Juliet and Zuma studies are reviewed here. No head-to-head comparison, and I guess what happened in at least my academic life, because there's no way they will do randomized study between T-cell, axi-cell versus lyso-cell. So we tried to compare with other methods. Now we can't get randomized study. We still need to find something to see how we can compare. Propensity score match analysis, PS. Inverse probability of treatment weighting, IPTW. Nice methods. Nice diagrammatic overview of both methods. Propensity score, inverse probability of Treatment waiting, frankly speaking, I don't understand this method as a clinician. My interest is outcome and recommendation. So let's review. These are matching variables. Slide review the patient characteristic of both group, TSA cell, 170 patient, XCell, 302 patient. Nice example of center imbalance adjustment. No residual significant difference in patient characteristics was observed for all other variables after stringent matching using either PS matching or IPT weighting approaches. You can see here best overall response and CR rate were significantly higher with XCell compared to TSA cell. There was no difference in DOR. Progression-free survival was significantly longer with XCell than TSA cell, 53% versus 32% at 6 months. Findings were confirmed in IPT weight analysis. Overall survival was no different between the group by both methods. All in numbers reviewed here for later review. Progression-free survival, best overall response rate, complete remission rate were superior after XCell, but no difference in overall survival and DOR between the group by both methods. Regarding toxicity, there was no significant different incidence of CRS, but XCell was associated with significantly more frequent and high-grade ICANS compared to TSA cell. In conclusion, I don't think I need to raise slide, very well known, and this is nice analysis to compare both products. So my favorite group of collaborators presented this superb abstract. Impact of molecular feature of diffuse large B-cell lymphoma on treatment outcome. Again, another study to predict which will not do better, which group of patients will not do better after CAR-T therapy. Primary therapy failure, we saw ALC and now molecular profile. Diffuse large B-cell lymphoma represents several distinct clinical pathological entities recently identified by molecular profile. Nice publication cited here, I would recommend strongly. We know that relapsed refractory large B-cell lymphoma has a poor outcome. To help this group of patients, we use CAR-T therapy, and this is their outcome. Treatment with anti-CD19 CAR-T therapy is now standard for many patients with relapsed refractory disease. But there are lots of patients who still fail to CAR-T therapy. The reason for failure after CAR-T reviewed here. Nice slide. In this study, authors sought to determine the impact of molecular feature of large B-cell lymphoma tumor on clinical outcome after CAR-T. Genetic analysis was done on 96 patients with pre-treatment biopsy, pre-CAR-T biopsy that passed sequencing quality filters and expression analysis of 93 patients. Baseline characteristics summarized here. Slide reviews overall survival after CAR-T infusion based on their response. Best overall response was CR in 43% patient and PR in 10% of patient. At that time of relapsed refractory disease, most commonly mutated genes are seen in the subgroup figure highlighted in the red, TP53, KMT, 2D, BP, BCL2, and other listed here. Inferior progression-free survival was observed in patients with mutation in BCL2, and see my lowest pink line you can see here. Progression-free survival by molecular subtypes at cluster is reviewed here. You can see EZB mutation and cluster 3, C3 had inferior progression-free survival. Progression-free survival by double or triple hit status, no difference and not bad after CAR-T. Unbiased model comprising mutation of MYC, BCL2, CDKN2A, and KLHL6 was strongly associated with lack of response to CAR-T therapy and poor prognosis. So in conclusion, larger series studying the impact of molecular feature of large B-cell lymphoma on treatment outcome of CAR-T. Double or triple hit component was not associated with adverse outcome after CAR-T therapy and consistent with previous report. Primary analysis suggested that mutation in KLH6, KLHL6, and CDKN2A, as well as MYC and BCL2, are associated with inferior outcome after CAR-T therapy. Nice abstract. Answer our most important question: what next for relapsed refractory CNS lymphoma? Can we use CAR-T? So this is a nice chapter. Small study, but very nice study. We don't have much data because primary CNS lymphoma is and was prohibited by FDA limitation of use for all three commercially available CAR-T products. And PCNS carries poor prognosis. Once patient relapse, extremely poor outcome in adult patient. There is a concern regarding increased risk of neurological events and lack of CNS trafficking in patients with primary CNS lymphoma after CAR-T therapy. So based on investigator's prior experience, smart guy, T-cell in secondary CNS, they conducted a pilot study with expansion of T-cell in adults with relapsed refractory primary CNS lymphoma. 12 patients received CAR-T for relapsed refractory primary CNS lymphoma. Patients' details are reviewed here. With median follow-up of 8 months for survival, 6 out of 9 patients relapsed refractory CNS lymphoma were alive, with 4 out of 9 showing ongoing response after CAR-T therapy. Responses were seen everywhere. Nice slide. Expansion of T-cell was demonstrated in peripheral blood and also in CSF. What about toxicity? No patient required intervention for CRS, but yes, ICANS developed in 5 out of 9 patients. Only a single case of grade 3 CR ICANS with median follow-up onset of 5 days. Remember, only a single case of grade 3 ICANS with median time of onset was 5 days. Very impressive. And this is an interesting course of patient who developed grade 3 ICANS. Patient received involved RT to tumor site to treat grade 3 ICANS with other modality of treatment. Nanostring and RNA pathway, which is famous nowadays, analysis of CNS demonstrate high degree of CNS CAR-T penetration in responding patient signature. Very nice figure. And increased T-cell and macrophage gene signature in responding patient. So in conclusion, T-cell in relapsed refractory primary CNS lymphoma was safe, efficacious in a highly refractory group of patients with significant unmet need. And look at the patient's characteristics. They were very highly, heavily pretreated, high-risk disease group. Common clinical topic, we always discuss reinfusing CAR-T in relapsed refractory B-cell ALL. Nice study presented by Regina. Author wanted to study effectiveness of reinfusion. Goal of reinfusion to mitigate the relapse risk, treat the relapse, and produce a response after non-responder to initial CAR-T infusion. 3 indications. Study design reviewed here for your later review. 81 received 1 or more reinfusion. Product details summarized here. Majority of patients received investigational CAR-T product as expected because this is a UPenn study. Prior allo transplant in half of the patient. My apology for this slide, some formatting issues again. Graph shows relapse and survival among patients reinfused for relapse prevention based on response to reinfusion. CIR, cumulative incidence of relapse, and overall survival were not statistically significantly different between patients with CR or no response at reinfusion. So save the time, this is a conclusion. Reinfusion of CAR-T is safe, may prolong B-cell aplasia in patients with short CAR-T persistence and reduce the relapse rate, and can induce remission in patients with CD19 relapse disease. Thus, reinfusion may provide alternative to transplant for short persistence. However, reinfusion is not effective for patients with non-response to initial CAR-T infusion. This is our last extract. Thank you for your patience. Interesting and important study. IACH prophylaxis with simvastatin and intrathecal dexamethasone in patients receiving axi-cell treatment. The component needed for severe IACHs are circulated. After when I received this abstract from Joseph, I asked him, I didn't understand why they use simvastatin for this prophylactic protocol. And this is what his explanation, and then I reviewed the literature. The component needed for severe IACHs are circulating cytokines and breakdown in blood-brain barrier that allows cytokines into the brain and CSF. Now, breakdown of blood-brain barrier is one of the component of endothelial dysfunction, and statins are able to reverse that. And that's the reason they selected statin for IACH prophylaxis. We don't need introduction. Severe IACHs require intense monitoring and therapy. Best we can do is prevent if it is possible. This is a feasibility study combining 40 milligrams simvastatin daily, starting at least 5 days prior to apheresis, in addition to dexamethasone, delivered intrathecally on day -1, +6, +13 in relation to axi-cell. Schema of trial is presented here. This is a registered clinical trial, clinicaltrial.gov. Numbers is listed here for your reference. Standard eligibility criteria listed here, nothing new. Investigators have accrued 6 out of 20 planned patients with relapsed refractory large B-cell lymphoma who received axi-cell per institutional guideline. Table shows the patient's characteristic. With a median follow-up 123 days, all patients achieved complete remission and remain alive. None of the 5 patients had grade 3 or 4 IACH. Only 1 patient had grade 1 neurotoxicity. Very impressive. Cytokine analysis and markers of endothelial and neuronal activation is underway. Impressed by the data. Friends, thank you very much for your attention, and please email me or call me if you have any question. Again, thank you so much.